So good morning, everybody, and welcome to the fourth webinar of the VALITEST webinar series on the organization of test performance studies. So VALITEST is an EU-funded project undertaken by a consortium of 16 partners and built to improve the reliability of diagnostic tests performed in plant health laboratories across the European and Mediterranean region. And you can find more information on VALITEST on our website. Test performance studies are interlaboratory comparisons that aims at evaluating the performance of diagnostic tests. And during this webinar series, partners are presenting the different steps of the TPS workflow through different videos, webinars, and training activities. So we already had several webinars on the preparation of the TPS plan, selection of the test, and selection of the participants. And today webinar, we will discuss about the preparation and dispatch of the samples. So the presentation will be recorded and a link to the recording and the PDF presentation will be provided on the VALITEST website. Also note that we will launch videos from the GoToWebinar platform during the webinar. And if your connection is not very stable, or if you are joining using the web app, the video can stop in the middle and not restart. So if you have this problem, please try to leave the webinar and come back, or watch the video on YouTube. I will provide a link for each video. Also, please do not hesitate to write in the chat in case you have any problem. So today, our speaker is Mathieu Roland. Mathieu is a member of the VALITEST coordination team. He joined the plant health laboratory of ANSES in France in May 2016 as deputy head of the unit coordination of reference activities. And before working at ANSES, Mathieu worked in the French Variety and Seed Study and Control Group, so GEVES, as head of the GMO and pathogens detection unit. So after the webinar, there will be a Q&A session. And for this Q&A session, we will be joined by 3 TPS organizers. So Natasha Mel, who worked at the National Institute of Biology in Slovenia and organized the TPS and TSWV. Jean-Philippe Renvoisé, who works for ANSES in France and organized the TPS on PPV. And Catherine Harrison, who works for FERA in the UK and organized the TPS on Fusarium cincinatum. So now we will start the presentation of Mathieu.
Thank you very much, Charlotte, for your introduction. So the presentation today is based on data provided by the TPS organizers of the project, but I will mainly focus on the examples of the TPS organized on Bursiophalanchus xylophilus by Anne-Marie Chappé from ANSES, and on the TPS organized on the plumpox virus by Jean-Philippe Renvoisé, also from ANSES. As Charlotte just said, we will also use some interviews of different TPS organizers, and at the end, we will save some time for a question and answer session. So just for those of you who may not have attended the previous webinars, a short reminder, VALITEST, as Charlotte said, is an H2020 European project. It aims at producing validation data for the detection and the identification of plant pests. And during this project, we organized 2 rounds of test performance studies. The first round was organized in 2019. It targeted 6 pests which had been identified by the consortium as important. On the slide, you can see the names of the different pests, you can see pictures of symptoms, and you can also identify the partners which have organized the TPS. Same for the second round of TPS organized in 2020, except that this time the pests were selected based on the needs expressed by various stakeholders. The idea was to try to align our validations with the needs of the stakeholders and with the market. So the organization of a test performance study is a complex and long procedure represented by this workflow you can see on the slide. And for today, we will only focus on the preparation and dispatch of samples and regions and on the stability and homogeneity studies. The presentation will be divided as follows. First, the scope of the validation, then the design of the panel, the production of the panel, and finally, the shipment of the samples. But first, the scope of the validation. So, before starting, a laboratory willing to organize a TPS should clearly define for each test and for each test the type of sample. It can be plant extract, wood, leaves, seed, fruit. It can be infected or non-infected plant material. It can also be plant material with deactivated pathogens, and it can be DNA or RNA extract. The organizer has also to identify the kind of matrix. It can be leaves, fruits, wood, seeds, and so on. It can be fresh or freeze-dried, and in some cases, it is possible to ship cultured pests. Before starting, it is important to identify if the test is suitable for symptomatic and/or asymptomatic tissues. It is important to identify the purpose. Will it be used for the detection and/or the identification? And even at the early stage of the preparation of the TPS, the organizers should think about the controls it will include. And you have here on the slide an extensive list of the different controls which can be used. And finally, the organizer should also determine the maximum number of participants and the maximum number of samples to use. This will be directly linked to the ability of the organizer to prepare all the samples, to ship them, but also to analyze the different results. So once the scope has been defined, the organizer has to think about the design of the panel itself and the composition of the panel. And to start this part, we will ask you two questions. In both cases, you will have some proposals on your screen and you can vote. We will give you 1 minute to vote. The first question is, how many samples, including the controls, you would include in a panel dedicated to a TPS? Would it be less than 10 samples, 10 to 20, 20 to 30, or 30 to 40?
So we have more than 80% of the people that have answered. I will wait a bit longer. Okay, so it's not really changing now, so I will close the poll and share the results. So we have 15% of the attendees that answered less than 10, 61% from 10 to 20, 18% from 20 to 30, and 6% 30 to 40.
Okay, so it's interesting to see that your answers do not exactly correspond to the guidelines we will provide in the framework of the project. It's more between 20 and 30, but I will give you more details in the next few slides. The second question we want to ask you is, according to you, what are the main parameters to define the type of samples to include? And we will ask you to select a maximum of 2 answers, and the proposals are the number of participants, the performance criteria to be assessed, the time and budget constraints, and the availability of plant material.
So we have about 80% of the people that have answered. I will wait a few more seconds. Okay, so now it's not changing much and the results are quite clear. So I will close the poll and share the results. So most of you, so 88% of the people and 79% of the people answered performance criteria to be assessed and availability of plant material. And then 10% number of participants and 6% time and budget constraints.
Yes, well, as we will see in the next few slides, I think it corresponds to what most of the TPS organizer would answer, even though the time and budget constraints are also extremely important when you prepare a TPS. Okay, so actually, when we first started to organize the TPS in the framework of the project, some of the partners had some experience in the TPS organization, but not all of them. And actually, we ended up with very diverse panel compositions. During the first round, we had panels with 8 to 22 samples, different number of dilution points, and even in some cases, no dilution points at all, and different number of replicates depending on the scope of the TPS. Of course, we had some difficulties to analyze the results, and these led to some bias in the test performance results. And we quickly realized that we needed a recommendation for the panel composition, and we needed these recommendations before the organization of the second round of TPS. And these recommendations had to focus especially on the composition and the number of samples, on the number of dilution points, and on the number of replicates in a sample panel. So this work was led by Sébastien Massard from University of Liège in the framework of the work package 2, improvement of the validation process. And on this slide, you can see a table which will be included in the guidelines provided by this work package. So what do we propose? We propose actually to have two biological negative samples with two replicates. It is important that these negative samples should be independent from each other. They should not be aliquot from a single negative samples. Same for the positive. We propose to have two biological samples with two replicates. We propose to have one sample with a medium concentration and another one closer to the limit of detection, but of course higher than the limit of detection. And same as for the negative, the positive samples should be independent from each other. Finally, we propose to have one serial dilution with the strongest concentration in duplicate and other dilutions in triplicate. We propose to have 5 dilution points with the highest dilution almost not detectable, and the positive sample used for the dilution series should be independent from the other ones, which makes a total of 3 positive samples. And a total of 22 samples. This does not include the controls. Standardizing the panel composition, of course, helps a lot to harmonize the validation studies, but it makes also the comparison of data much easier and actually more robust. Using this data, it is much easier and you can really trust the results you obtain when you want to compare different tests. However, it is important to keep in mind that these recommendations correspond to a compromise between the statistical requirements and the practical constraints. There is no obligation. Panel composition depends on the performance criteria to be assessed for the test. And it actually may not be feasible in some cases due to the number of reference material or the absence of biological material. And here we clearly see that it corresponds to the votes you just had for the previous question. Now we propose you to see a few interviews of TPS organizers, and the question we asked to TPS organizer was, what was the composition of the panel of samples?
What was the composition of the panel of samples?
Composition of the panel of samples was designed in such a way that it improved our knowledge of the specific tests and host plants included. So for example, for Erwinia amylovera, we know that there's a lot of data on apples, but we also wanted to generate new data on pears, on certain ornamental plants, and we've also included those hosts in the test panel. While for Pantoia stevarti, we know that the challenge is to be able to detect low levels of the pathogens and to differentiate between the two subspecies that can occur in such samples, subspecies stevarti and subspecies endogenous. So in that case, we've had samples spiked with low levels of the target, and also some samples containing subspecies endogenous, which is cross-reacting in many of the molecular tests for detection.
The panel was composed of 11 samples in duplicate for a total of 22 samples. We included 5 negative samples in duplicate, 5 positive samples in duplicate, each belonging to a different isolate and strain of the target, and 2 positive samples in duplicate corresponding to 2 deletion levels of one of the previous isolates.
The composition of the panel of samples was, we had in total 16 samples. We had a panel of 8 DNA extracts and a panel of 8 spiked wood material.
The panel of sample was prepared in accordance to the guideline of the Work Package 2 teams and TPS Round 1 organizer. Specifically, 9 samples were included in the panel, some in double and some in triplicates. The panel included leaves and fruits of tomato and pepper plants and was composed of 2 healthy samples, 1 infected samples at high level of dilution, 1 at low, 1 at medium concentration. For a total of 22 blind test items. We also provide the isolation and amplification controls.
So, as you will have seen in this video, many organizers did follow the recommendations, but not all of them. And we actually had good reasons to do so. Just 2 examples. The first one on the tomato spotted wilt viruses, a TPS organized by NIB. You can see that the recommendation is to have 4 negative samples, and for the conventional PCR we had 8, and that's ELISA we had 12. Actually, as it's been said in the video, NIB knew that there are some cross reactions of some TSWV specific tests with other similar TOSPO viruses species, and it made sense to collect more data concerning the exclusivity. And it's the reason why they decided to include other TOSPO virus isolates in the panel. For the PPV this time, you see that we recommend to have 14 samples in the dilutions, and the panel included only 4. Actually, in this case, the tests used were on-site tests, and on-site tests are mostly dedicated to symptomatic samples. However, we can't tell if diluted samples are similar in terms of concentration to symptomatic samples. And it's the reason why the analytical sensitivity was not specifically assessed. And the organizers decided to focus on the analytical specificity and on the inclusivity especially, but also on the repeatability. So once the panel has been designed, we can talk about the description of the different types of samples. And once again, we will show you some interviews of the TPS organizers. This time the question asked was, which samples were used to evaluate which criteria?
Which samples were used to evaluate which criteria?
So with both test performance studies, we were mostly focusing on analytical sensitivity, analytical and diagnostic sensitivity of the tests, but we had some specific samples, as I mentioned, for example, for Pantoes-Tavarti, samples that did cross-react and could give us information on both on analytical specificity and diagnostic specificity of the different tests. In all TPS studies, we have also included controls at different levels. So in all our cases for test performance studies on molecular tests, participants also did DNA extraction, and we have provided controls for both DNA extractions and for molecular tests themselves. And these were basically the results that we used to judge whether the data sets the participants submitted were fine or not. If the controls did not have the correct results, the data sets were excluded.
So the main criteria we determined were the concordance or accuracy. So this means is to what extent were the results in agreement with the expected outcome that we determined in our preliminary studies. And furthermore, we looked at diagnostic sensitivity and specificity. And the first one, the diagnostic sensitivity, refers to the test ability to detect the target. And in case of sensitivity, we are talking the ability to not detect the non-target. So those were the three main criteria we looked at.
So we use target samples to evaluate diagnostic sensitivity and non-target samples to evaluate the diagnostic specificity. And the positive samples at different level of concentration were used to evaluate the probability of detection and the accordance, also called repeatability for qualitative tests, and concordance, also called reproducibility for qualitative tests.
So coming back to the presentation. Yes. So most of the comments of the TPS organizers can be summarized in this figure prepared by Sébastien Massard in the framework of the work package 2. And in this figure, we can see that actually LC samples and infected samples are used to assess the diagnostic sensitivity, the diagnostic specificity, other performance criteria, and the associated confidence intervals. The diluted infected samples are used to assess the analytical sensitivity, and all the samples are used to assess the repeatability and the reproducibility. If we focus on one example, the PPV, in this TPS, we used healthy sample, 4 different matrices in 2 replicates, to assess the diagnostic specificity, selectivity, and repeatability. We provided infected samples, actually 5 different strains and 2 replicates to assess the diagnostic sensitivity, the inclusivity, and the repeatability. We provided diluted infected samples to assess the analytical sensitivity and the repeatability. And finally, 11 datasets were used for statistical analysis to assess the reproducibility. Now that we've designed the panel, we can move to the production of the different samples. And first, we have to access the biological material. Once again, we will show you an interview of different TPS organizers. The question asked was, what was the availability of the biological material and what were the difficulties to get and collect the material?
What was the availability of the biological material and what were the difficulties to get or collect the material?
Xylopholis ampolinus is a pathogen of vines. In the UK, there was no vegetative material present over the period of the TPS, so we had to use vine stem material. As you can imagine, this is a particularly tough material, so in order to produce a homogeneous sample, we had to try a number of homogenization methods to be able to grind the stem up enough to produce large quantities of lysate. The method we found most effective was to cut the stem material up using secateurs and place these pieces, small pieces, into large Nalgene bottles containing large ball bearings with a small amount of buffer. We then placed these samples on a paint shaker machine and ground them for just over 10 minutes, adding additional buffer halfway through the grinding process. Samples were later spun and a clear lysate produced and pulled into a single batch ready for spiking with Xylopholis ampolinus cells.
So we were lucky because a lot of isolates for tospoviruses are in DSMZ collection. This is German collection. In addition, do a good collaboration with many laboratories around the world, we were able to collect many different isolates.
For Plumppox virus, we maintain a couple of isolates in our own collection, but furthermore, we ask different laboratories and institutions to share their isolates, and we received isolates from collections from Hungary and Italy and France, Russia, and the Netherlands. So we didn't really encounter any problems in receiving extra isolates, and it went pretty well actually.
After the first outbreaks in Italy, infected material was provided to our laboratory for confirmatory tests. This allows us to have different local isolate as leaves, fruit and seeds, which were maintained dehydrated or frozen. All those isolates were artificially inoculated on tomato and pepper plants to increase the amount of biological material available for pretest and TPS. The amount of the obtained infected plants was sufficient for preparing all the reference material to be distributed among the participants. Other tobamovirus used for testing specificity were bought at DSMZ.
We have a collection of Burcephalanthus that we use as controls in routine analysis of our validation test. Burcephalanthus grow very well on Botryoptinia in Petri dish, so it wasn't difficult to find material for this TPS.
It wasn't possible to source naturally infected material for the test performance study, so we ended up just using pure cultures and extracted DNA. And we also decided not to include, not to spike material because of difficulties in the type of host material. So for this test performance study was purely pure cultures and DNA that was dispatched and no host material, just because of the difficulties in the matrix and being able to source naturally infected material.
So, as you will have understood from the comments of the TPS organizer, collecting the biological material is not always an easy task. Just one example about the PPV. Of course, having fresh material to organize the TPS is the best option, but it is not always possible to collect and test the material when you want to organize the TPS. And it was the issue with the PPV. We had to follow the time frame of the project, and we could not organize it while the biological material would have been available. Another option is to deseasonalize the production in dedicated facilities, but again, it is not an easy task. Last option is just to collect and store the material, and then, of course, you have to check for potential matrix effect. You have different storage solutions. You can freeze dry, extract nucleic acids. Using on-site tests, you want to collect the recommended matrix. We will not evaluate a test on a different species or a species which is not meant to be tested using this assay. And you want to avoid potential matrix effect. We wanted to collect symptoms for target and non-target pests. And again, you want to do it at the best period. It is easier when you want to collect healthy material. This was not an issue. And same for the dilution. You just have to dilute some of your samples to evaluate the analytical sensitivity. So we said that the access to the biological material can be difficult. In some cases, you can also multiply it. So we already talked about the collection of the material. We have to respect the seasonality. You have to be careful about the way you will store your material. You have here a picture of some citrus leaves collected for the evaluation of tests on the citrus tristeza virus. In other cases, you can also produce your biological material. It will depend on the pest, and some pests cannot be cultured or only with difficulty. And you also see some illustration of here petri dishes with the fungi Philosticta citricarpa and greenhouses for the maintenance of nematodes on plants. An example in nematology with Bursophalanchus dilophilus. Actually, this nematode can be raised on fungi culture. So what is done, as shown on the pictures, is that we grow the fungi, in this case, a non-spirulent strain of Botrytis cinerea. And once you have the fungi, you can raise the nematode on your petri dishes. Once you have your nematode, you can just collect them and use them to spike, for example, wood, like shown on the picture on the bottom of the slide. Once you've produced your samples, you have to check the status, the homogeneity, and the stability. So, of course, results should not be influenced by the preparation of the samples, and we really want to avoid an uneven distribution of the pest in the matrix. And it's the reason why homogeneity is a crucial point to be met. So, to do so, you have to test samples for your homogeneity. And to do it, you have to take a few decisions. First, which test should be used for the homogeneity evaluation. Then the number of units you want to test for each production lot, and the decision rules you will apply to your results. It is also a good time to determine the assigned value of your samples. And of course, you will only choose the homogeneous samples to constitute the different panels. You also have to think about the stability of your samples. To do so, you have to store dedicated samples in the same conditions as for the participants. You have to set a short-term deadline for analyzing the samples. You have to test the samples using the same test as for homogeneity. And again, you have to take almost the same decisions, like the number of units you are willing to test and the decision rules you will apply to your results. And if some of the lots are not as stable as expected, the results associated to these lots should not be taken into account for the statistical analysis. So just a short advertising for the 5th webinar of our series. Most of these points about the production of material will be presented in more details in the webinar on the production of reference material for TPS, which will be presented on Wednesday the 10th at 2 p.m. We can now move to the shipment and first of all the coding of the samples. So of course you want to randomize your sample. You want to do it to ensure a fully blind testing of the samples. You need individual codes of samples randomly chosen. You need to choose different names, different identifications for each panel and for each participant. And on this slide, we just show you an example. It actually corresponds to the stickers which were on the tubes and on the bags shipped for the PPV on-site testing. PPV2P04B18 stands for PPV2 corresponds to the TPS code, P04 to the code of the panel, B corresponds to one of the tests, and 18 to the code of the sample. The regulatory requirements are important when you want to ship some material. In the case of Bursaphelancus xylophilus, we wanted to to assess different tests on different methods, and we shipped different kind of biological material. To assess the tests on real-time PCR and LAMP, we shipped DNA extracts and wood extracts spiked with nematodes. So in this case, we shipped living material. And of course, this needed a letter of authority or an import permit according to the country where we wanted to ship the samples. In the case of conventional PCR, we just sent DNA extracts, and in this case, there was no requirement. So during the project, we prepared a lot of different documents, including the participants' contract. And in these documents, it is clearly stated that regulatory documents should be received before sending the samples. And if it is not the case, the legislation will not allow the organizer to ship the parcel to the participant. And actually, the participation in the TPS will be compromised. So again, this is an important point. Finally, the transport and administrative documents. On this slide, you can see some illustrations and the composition of what was shipped to the participants. So in the case of Bursaphelancus xylophilus, we shipped mostly DNA extracts in microtubes, but also some spiked wood extracts, plus, of course, the primers, the probes, and the controls. In the case of PPV, we mainly shipped freeze-dried leaves in tubes or in bags, but also some specific panels for tests requiring specific bags and buffers. So as I said previously, during the project, we had to prepare a lot of different documents, including the TPS technical sheet describing the experimental protocols to be performed, the TPS instruction sheet describing the instructions to be followed at each step in the implementation of the TPS, the acknowledgement of receipt of the sample, and the TPS result form to record the TPS results. Last video of interviews of the TPS organizers. This time the question asked was, did you encounter any logistic or regulatory problem?
Did you encounter any logistic or regulatory problem?
Yes, so the regulatory status of Xiphilis ampellinus changed sort of prior to the TPS. In England, we no longer required a letter of authority to receive Xiphilis ampellinus samples. However, a number of the participant countries still did when they checked with their plant health regulator. Also, obviously, the lockdown had a big impact on the completion of the testing within the time frame and resulted in one of the participants dropping out, unfortunately.
Yes, our panel of samples included also one quarantine virus. This was Chrysanthemum stem necrosis virus. Therefore, letter of authority was required. I must say that some labs had big problems with getting the letter of authority in time. And I think that one lab was even not able to get it, most probably due to COVID epidemics.
To ship all the samples, a letter of authority was required. Unfortunately, not all the selected participants were able to have it in time. The emergence COVID-19 exacerbated this problem. We sent everything in dry ice, but it was some logistic problem to send the material to New Zealand, and we circumvent this problem by shipping samples at room temperature. At the end of the TPS, all the participants are we are able to send materials and reagents to all the participants.
Some laboratories struggled to get their letters of authority in time for the planned dispatch date, so some samples were dispatched a little bit later than was originally planned, and I think one laboratory we just dispatched DNA to them because they couldn't get their letter of authority approved.
So, which lessons have we learned while organizing all these TPS? Of course, we had difficulties, especially we had issues with the seasonality of plant material. We had to collect plant material in advance. We had to store the material and be extremely careful about the stability of the stored material. We had difficulties to collect enough biological material. Sometimes we had to infect artificially some hosts, or we had to culture and extract DNA. We had some difficulties to find effective homogenization method, especially with some tricky matrices such as wood. And finally, as you've heard in the video, we had a lot of difficulties to get the LOA on time. So what about the improvements? We realized that we needed more time between sending the contract and sending the samples. We needed more time to order the reagents, but also to get the LOA. And it's the reason why we decided to extend this period of time from 1 to 2 months during the between the two TPS rounds. So in conclusion, actually, some of the partners already had experience in organizing TPS at the beginning of the project, but we've all learned together how to improve this process, and it is the reason why today we are willing to share this experience. But I think the best conclusion we can give is that during the project, around 9,700 samples have been prepared and dispatched worldwide. So, of course, I could not go into too much details during the presentation, but we have a question and answer session just afterwards. So feel free to ask any question you would have. Now is the time to ask your question to people who truly were involved in the organization of this TPS. Thank you very much for your attention. And for the questions, I think you can ask them directly in the chat. Or maybe raise your hand. I don't know, Charlotte.
Yeah. So I will ask, so thank you, Mathieu, for the very nice presentation. I will ask the TPS organizers to join us. So turn on their camera and their microphone. And yes, you can ask your questions either in the chat, or you can also raise your hand and we will give you the floor if you have a question. And so for now, I don't have any questions. So maybe if Natasha, Catherine, or Jean-Philippe have any reaction on the presentation that was just made by Mathieu, they can react, and I will wait a few more minutes. So yeah, really do not hesitate to ask questions to the TPS organizers.
Actually, I think that there is nothing to add because Matthew already described all our problems that we faced on. Yeah. So thank you, Matthew.
Maybe just one point about the different documents which were prepared. Maybe we could say that they will be made available, at least in the booklet we will publish in Springer. So I don't know when this booklet will be available, but at some point, all the documents we've been preparing will be made available for the community.
So I still do not have any question from participants. Wow. So Mathieu, it was very, very clear, I guess. So yes,
well,
I have a comment that is, it was quite interesting to see your side as organizers. Thanks. So it's not really a question, but okay. Well, so
if there are no questions, thank you for attending.
Yes, so I thank you, Mathieu, and thank you to the TPS organizers for being there, although there was no question. Oh, I have one question. So in the case of viruses, TPS, how do you guess a low titer positive control?
Yeah, actually, in case of viruses, this is not easy to estimate. You can, let's say, count the number of RNA targets or DNA targets using droplet digital PCR assays. What we did is, in that case of the metaspotted white virus, is that we prepared a dilution series. We tested this dilution series. And you can imagine that for different kind of the methods, the low concentration of the virus was different. This means that in case of real-time PCR, we included a low positive control, higher dilution as in case of ELISAs and on-site tests, because of the differences in the sensitivity. So this is just the estimation making the dilutions that you can do for viruses.
May I can say something? In the case of the mpox virus, we used an RT-PCR to test for homogeneity, and of course we have already tested all our samples previously, so we had an idea about the CQ values for all positive controls, positive samples, and of course all the dilutions we have prepared.
Thank you both for the answers. Okay, so now the question are coming. So what if the sample don't arrive at good stage? Are they sent again, or just the TPS is stopped for that lab?
If I may start, actually what we did is that we prepare some batch of samples for such cases. So if this would happen, then we have some batch of samples to send to the labs again. In that particular TPS, it doesn't happen, but yeah, it is possible. So we always prepare some batches more for such cases. And we hope that this will not be a case for many labs.
We have prepared 2 extra panels just ready to be sent.
Okay, and there is another question regarding LOA. Is it possible to arrange it yearly to avoid the problem of not having it in time when needed?
I think that this is depend on the country, depend on the National Plant Protection Organization, how they would like to do this. So in our case, currently this is not possible. We need to ask for each study separately to clearly specify the number of the samples we will get, the time when we'll perform the analysis. But I guess that this is maybe different in different countries.
Yeah, I think that it's difficult to have a yearly one for a test performance study, because you're going to do different sample types, and you're not going to do the same study more than once in the year. So I think that would be a difficult thing to do. Maybe if you were doing reference material and sending it out regularly as more of a proficiency test, then that might be more applicable.
Okay, so we have no more question. Yes, thank you again all. Thank you to the attendees for participating to the webinar. There will be a satisfactory survey at the end of the webinar or in the follow-up email. It is really short, so do not hesitate to provide your feedback on the webinar. And as I said before, the presentation and the link to the recording will be available on the website as well as on Twitter. And finally, the next webinar will be on the production of reference materials. So it will be a presentation of the work done by WP3 on the guidelines on how to produce reference material. And it will be next Wednesday at 2 p.m. So thank you again to everybody for attending, and see you at the next webinar. Bye-bye.
Thank you.